Hydra habit, habitat, external morphology, internal structures. Hydra are inconspicuous freshwater relatives of corals, sea anemones, and jellyfish that are classified under the phylum Cnidaria, class Hydrozoa, order Anthomedusae/Anthoathecata, and family Hydroidea. It is essentially a sedentary organism living attached to stones, pebbles, and water plants, but can get released from the substratum and swim. The name Hydra was given by scientist Linnaeus because of its special power to regenerate its lost part, like Hydra, a nine-headed dragon serpentine of the Greek mythology. Systematic position. Phylum, Coelenterata. Class, Hydrozoa. Order, Hydroidea. Suborder, Anthomedusae. Genus, Hydra. Habit and habitat. Hydras are solitary, sessile, freshwater animals, cosmopolitan in distribution. They can be found in a large variety of freshwater habitats, mostly occur in ponds, lakes, and slow-flowing parts of streams and rivers. They may be found attached to and hanging downwards from the underside of solid objects in the water such as leaves, sticks, stones, weeds, etc. The tentacles and body of Hydra are stretched to the maximum limit in water in order to capture any prey that comes in contact with them. When disturbed, the body at once contracts into a minute jelly-like knob. They are carnivorous in habit and feeds on insect larvae and, especially, small crustaceans such as water fleas, seed shrimps, and copepods. Collection and preservation. Hydra can be collected from shallow water ponds, lakes, and streams during early winter. As these animals live habitually clinging to aquatic vegetation, quantities of vegetation may be picked and put in a jar filled with pond water. In the laboratory, hydras may be dislodged from vegetation by squirting them with a jet of water from a pipette. They are then transferred to a dish with a pipette or small aquarium net. When fully expanded, hydras may be fixed in Bouillon's fixative for 30 minutes, given several washes with 50% alcohol, kept in 50% alcohol for 10 to 15 minutes, and finally preserved in 70% alcohol. External morphology. 1. Shape and size. Hydra is a polyp-like or polypoid coelenterate with a tubular or cylindrical body. It becomes elongated and slender and measures about 1 centimeter in length when fully extended. When retracted, the body becomes shortened and somewhat globular and measures only a few millimeters. The symmetry of the body is typically radial, comprising an aurola-boral axis, with different parts arranged concentrically around it. 2. Coloration. H. vulgaris is colorless, H. gangetica is white or pink in color, H. oligactus and H. fusca which is brown in color, and H. viridissima which is green in color, green hydra. Asterisk the green coloration of H. viridissima is due to the presence of zoochlorelli, a unicellular green alga, that lives in symbiosis with the hydra. 3. Petal disc. The proximal or arboreal end is termed as a petal disc, is flattened and plays an important role in temporary attachment. It consists of the glandular zone involved in the secretion of adhesives that make it possible for the organism to attach to the substratum and also produce gas bubbles that allow hydra to float. 4. Hyptostome, mouth, and tentacles. The distal or oral end of the body column consists of a conical hypostome. Hyptostome contains a circular aperture or mouth at its apex, which opens into the gastrovascular cavity or enteron. Hypostome is surrounded by 6 to 10 slender, contractile, and tubular thread-like processes, called tentacles, L-tentar, to feel, which can be stretched to several millimeters to capture prey. These tentacles are controlled by a simple neural net. Tiny stinging cells, called nematocysts, cover the tentacles and are very specialized, and engage in offense and defense, playing a role in feeding by paralyzing the prey. Tentacles assist organisms in feeding and locomotion. Asterisk stinging cells or nematocytes of cnidarians represents one of the most poisonous and sophisticated cellular inventions in animal evolution. 5. Buds. The external surface of the hydra in some individuals may bear proximally lateral buds in various stages of development. Hydras reproduce asexually, budding. Budding results in the rapid production of a large number of genetically identical hydras. 6. Gonads. Gonads may be found as projections from the external surface of the body during the breeding season. When present, the male gonads or testes are found at the oral end as conical projection, whereas the single female gonad or ovary occurs near the basal disc as oval projection. Internal structures. The internal or histological structures of hydra are best seen in its longitudinal and transverse sections. Gastrovascular cavity. The internal structures reveal the presence of a body wall and a central cavity or coelenteron, or koilos, hollow plus enteron, gut, functionally referred to as the gastrovascular cavity. It is surrounded by the body wall. The mouth leads into this cavity, which extends into tentacles as their lumen. There is no anus and no excretory pore. Body wall, histology. Hydra is a diploblastic animal, i.e., it is derived from two germ layers, the ectoderm and endoderm. These germ layers form two distinct cellular layers, the outer epidermis and the inner gastrodermis, respectively of body wall and tentacles. The body is a hollow tube consisting of two layers of cells, separated by a thin, delicate, transparent, and non-cellular mesoglia. A. Epidermis. It is composed of small, more or less cuboidal cells. It forms a thin layer, about 1/3 of the thickness of the body. It acts as the protective and sensory layer and is enveloped by a thin coating of the cuticle. It's composed of epithelial muscle cells, gland cells, interstitial cells, cnidoblasts, sensory cells, nerve cells, and germ cells. 1. Epithelial muscle cells. Roughly conical or pear-shaped epithelial muscle cells have both epithelial and muscular parts in the same cells. The outer epithelial part extends up to the body surface, while the inner or basal muscular part is drawn out into two muscle processes along the longitudinal axis of the body. The muscle processes contain a contractile fibril myoneme or unstriped muscle fiber. The ectodermal myoneme run parallels to the long axis of the body and tentacles. They form longitudinal muscle, which brings about contraction of the body. These cells have a large nucleus, the row of granules along the border which secretes the cuticle. The epidermal cells of the basal disc are granular and they secrete mucus for the attachment of hydra. The basal epidermal cells also can form pseudopodia by which the animal glides on its attachment. Some granular epidermal cells of the basal disc secrete a gas to form a bubble by which hydra breaks from its attachment and lift it up. Electron microscopy has revealed the detailed structures of epitheliomuscle cells. In addition to the presence of usual intracellular organelles, like the nucleus, Golgi bodies, mitochondria, endoplasmic reticulum, ribosomes, lysosomes, and vacuoles, there are some other organelles as well. The cell membrane at the outer free surface has a few outward projections or microvilli. At the periphery below this membrane are present a few mucous bodies. These secrete a finely granular material to form a thick, protective mucus layer on the cell surface. The base region of the cell lies above the mesoglia and has many muscular processes, which run parallel to the long axis of the animal body. These muscular processes are filled with myofilaments, which constitute myoneme. Functions. They form a protective covering of the body. They help in contraction, shortening, and bending of the body. They help in locomotion. They help in attachment with a solid object, and they help in respiration through the mucus layer at the cell surface. 2. Gland cells. These are tall cells found abundant in tentacles, oral region, and pedal disc. Functions. These secrete a mucus-like sticky material that serves for attachment, protection, and entanglement of prey. Sometimes they secrete a gas bubble by which Hydra can rise and fasten onto the surface of the water to float. 3. Interstitial cells. These cells lie between narrow basal ends of epitheliomuscle cells, hence known as interstitial cells. These are small, rounded, undifferentiated embryonic cells that measure about 5 in diameter. Each cell contains a clear cytoplasm and a large nucleus with one nucleolus. Interstitial cells form a growth zone just below the tentacles. From this zone all kinds of new cells arise which push out the old worn-out cells, which are shed at the proximal and distal ends. These cells are capable of developing into any kind of cell such as reproductive, glandular, stinging and buds, etc., as required. They are thus totipotent or reserve cells. All the cells of Hydra are replaced by interstitial cells every 45 days according to Brain, 1955. Electron microscopy also reveals interstitial cells are small, rounded, or oval, about 5 in diameter. The cytoplasm is filled with the smooth endoplasmic reticulum, scattered ribosome, and a few small mitochondria. The central nucleus contains scattered granules. The nucleolus is smaller or absent. Functions. These cells are the main agent in rebuilding tissues during growth, budding, and regeneration. They form gonads during the breeding season to give rise to germ cells. They are capable of forming cnidocytes. 4. Cnidoblasts. Cnidoblasts, are weapons of offense and food capture. Many of the interstitial cells of the epidermis become specialized to form stinging cells, called cnidoblast, gr, cnide, nettle plus blastose, germ. They are found mostly on the tentacles and arranged in groups or clusters, batteries. A cnidoblast is somewhat oval with a basal nucleus and contains the sac-like organoid, the nematocyst or stinging cell. Nematocysts are characteristic of Cnidaria. The stinging cage contains a capsule with a coiled tube or thread. A sensitive hair looks out on the cell surface. When the victims of the Hydra swim by and touch the hairs, a stinging thread shoots out of the cage. In some stinging cells, the threads pierce the cover of the arthropod, in others, they inject poison inside, in others, they stick to the victim. Functions. These form organs of offense and defense of Hydra. They also help in food captures, locomotion, and anchorage. Nematocysts. One of the most characteristic structures of all coelenterate are the stinging cells, called nematocysts. Nematocyst, minute, elongated, or spherical capsule, produced exclusively by members of the phylum Cnidaria. These are organelles found in specialized cells called cnidocytes or cnidoblast. These are not a cell, because it is chitinous and non-living. These cells develop only from modified interstitial cells of the epidermis and are not found in the gastrodermis. When fully developed, cnidoblasts containing developing nematocysts migrate to tentacles through mesoglia by means of amoeboid movement. Projecting in between the epitheliomuscle cells, host cells, the cnidoblasts act as organs of offense and defense. They also serve locomotion, food capture, and anchorage. Structures of a cnidoblast. It is an oval or rounded cell with a conspicuous basal nucleus lying on one side. The nucleus contains a small inconspicuous nucleolus. The interior of the cnidoblast is filled by a peculiar oval or piriform sac or bladder, the stinging capsules, or nematocyst, gr, nema, thread plus kystis, bladder. Nematocysts consist of a tiny bulb or capsule, 5 to 50 in length, and composed of collagen-like protein. The capsule is filled with a poisonous fluid, or hypnotoxin, which is a chemical mixture of proteins and phenols. The narrow outer end of the capsule is invaginated into a long, hollow, tubular filament or thread tube, coiled like a watch spring inside the sac itself. The base of the threaded tube is swollen to form the butt or shaft. Inside the butt are 3 large spines, called barbs or stylets, and 3 spiral rows of minute spines, called barbules or spines. The bud is covered externally by a little lid-like structure, operculum. The outer narrow edge of the cnidocyte projects freely beyond the epidermal surface, as a tiny, pointed and hair-like process, the cnidocyl, gr, cnide, nettle plus cilium, hair, or trigger. Upon contact of the cnidocyl with some small animals causes sudden contraction of the cytoplasm, the nematocysts are discharged firing a dart-like thread containing neurotoxins. The cytoplasm of the cnidoblast may contain contractile muscle fibrils. In some, a restraining thread, called a lasso, is attached to the base of the cnidoblast. It prevents the nematocyst from being thrown out of it. Electron microscopic studies have shown the presence of endoplasmic reticulum, free ribosomes, basally located Golgi bodies, mitochondria, and multivesicular bodies in the cytoplasm. A bundle of small myofilaments, probably lasso, is present in the basal region of the cnidoblast extending from the capsule of the nematocyst. The cnidocyl is composed of a central core surrounded by large 20 to 21 hollow one rods. The core is structurally like a cilium as it contains fibers in 9+2 pattern. Fine microtubules are attached to the base of the capsule, which is double-walled. 2. Occurrence of nematocysts. Nematocysts scattered, mostly singly, throughout the epidermis of the body but remain absent on the basal disc. They are especially abundant in the oral region and on tentacles, where they form nematocyst batteries. A battery consists of 1 or 2 large central nematocysts, surrounded by 10 to 12 smaller ones, all enclosed within a single large epitheliomuscle cell. Cnidoblasts are never formed in tentacles where they literally migrate from their seat of origin in the epidermis of the body, or they may enter the body cavity. Hence they are passively transported in large quantities to the tentacles where they insist in the cluster to become batteries. None seems to insist in the gastrovascular cavity. 3. Mechanism of discharge. When the cnidocyl is stimulated by food, prey, or enemy, the explosion or discharge of nematocyst takes place. Neither touch alone, nor the presence of food together causes discharge, but touch and the presence of food together cause it. Both mechanical, such as contact with prey, and chemical stimulation, emanating from an approaching prey, are involved in the mechanism of discharge. The exact nature of discharge remains unknown. The response is wholly local and not under the control of the nervous system. The enzymes involved in discharge also remain unknown. It is not known why the well-fed Hydra fails to discharge nematocysts in the usual manner. The wall on nematocysts remains impermeable to water except at discharge. On the stimulation, the capsule wall suddenly increases its permeability. This causes a rapid intake of water and greatly increases osmotic pressure inside the capsule. As a result, the lid of an operculum is forced to open, the coiled thread tube turns inside out, and the entire nematocyst explodes to the outside. The contraction of contractile microtubules surrounding the capsule help in the discharge of nematocyst as attributed by some workers. Probably the neuronal connections bring about the coordinated discharge of nematocysts. As the threaded tube everts, the barbs and spines present inside the butt unfold to the outside. Thread tube once discharged cannot be withdrawn, so that exploded nematocysts cannot be used again. Their cnidoblast migrates to the gastrovascular cavity and are digested. The discharged nematocysts are replaced within 48 hours. 4. Types of nematocysts. Hydra has 4 types of nematocysts serving different functions as follows. A. Penetrants, stenatiles. B. Volvents, desmonemes. C. Steriline glutinant or Atricus isories, small glutinants. D. Streptoline glutinant or Holotricus isorises, large glutinants. A. Penetrants, stenatiles. It is the largest and complex nematocysts, which are about 16 m. It is spherical, occupying almost the entire inner space of the cnidoblast in which it lies. Its thread is long and the hollow tube remains coiled in a transverse plane. The thread bears 3 pairs of large stylets and 3 rows of small thorns or nettles at its stout base. When discharged, thread pierces the animal body and injects the poisonous fluid hypnotoxin, which paralyzes the victim or kills it outright. Hydra then seizes its prey with tentacles and draws it into its mouth. Bivalvence, desmonemes. It is small 9 m in size and pear-shaped nematocyst resembling a miniature bola. It has no butt. It contains a thick, spineless, smooth, and elastic thread tube forming a single loop and closed at the far end. When discharged, the thread coils round small projections such as hairs or bristles of the prey, thus impeding its movements. They are also useful in capturing the prey. C. Sterylene glutinate or Atricus isoris, small glutinants. It is oval or elongated nematocysts of about 7 m in size. It has no butt. Its thread is open at the tip and has no spines. They discharge a straight and unarmed thread tube pen at the tip and used for attachment. D. Streptoline glutinate or Holotricus isorises, large glutinants. It is oval or cylindrical, which is about 9 m in size. Its thread long with a narrow butt and forms 3 or 4 transverse coils. It is pointed and opens at the tip. The thread coils round the prey after discharge. It bears a spiral row of small spines and may coil upon discharge. Glutinants secrete a sticky substance possibly used in locomotion by fastening the tentacles of Hydra to solid objects, as well as helps in food capture. They also use to impede the progress of small animals when the projectiles are stuck to their appendages. 5. Sensory cells. They occur scattered throughout the epidermis among epitheliomuscle cells, especially on tentacles, hypostome, and petal disc. These are tall, narrow, and columnar, thread-like cells, usually bearing a delicate hair-like process, apical cilium, at their outer free tips. Their bases are continuous into 1 or more fine fibrils passing into the general nerve plexuses. They serve as undifferentiated receptors for sensitivity to touch, temperature, chemical stimuli, and light e.t. Functions: a sensory cell acts both as receptors and as a sensory neuron. That is, it both receives and transmits impulses. Electron microscopy reveals that the apical hair-like process is in fact a cilium arising from a notch at the apex of the sensory cell. The cilium consists of 9 peripheral and more than 2 central microfibers arising from a basal granule from which small rootlets extend into the cytoplasm. The apical cytoplasm contains mitochondria and small vesicles, and the microtubules extend into the apical collar. A Golgi apparatus lies above the nucleus. The basal end of the cell is either situated above a ganglion cell or gives rise to a process. 6. Nerve cells or ganglion cells. They are derived from the interstitial cells of the epidermis and are small or elongated. They occur at the base of epitheliomuscle cells just above their muscle processes, forming nerve net or nerve plexus. Each nerve cell consists of a small cell body containing the nucleus and gives off to several nerve processes or neurites, which are not differentiated into dendrites and axons as found in higher animals. The neurites of adjacent nerve cells from synaptic contacts, i.e., they lie very close together with microscopic gaps between them. Nerve cells inked up with synaptic contacts thus form an epidermal nerve net throughout the body of Hydra. The nerve net conducts impulses equally well in all directions due to restricted or no polarization. Electron microscopy of nerve cells shows the irregular plasma membranes with numerous crests and indentation. A small, oval nucleus surrounded by nuclear membrane-bearing pores. A smooth and rough-surfaced endoplasmic reticulum is present, but it is not a prominent feature. Mitochondria may be scattered or clustered. Golgi apparatus is most prominent and 2 or 3 separate Golgi regions may be present. Golgi apparatus is usually situated between the nucleus and a longitudinal process or neurite extending from the nuclear region. The cytoplasm also contains a number of small and large vesicles. The cells have highly developed microtubules that extend long distances in the neurites. But the nerve cells of the basal region of Hydra are devoid of microtubules and poor in ribosomes. The neurites may be longer than 10.0 and contain ribosomes, small and large vesicles, mitochondria, and microtubules. The neurite is lined by the plasma membrane of the neuron. 7. Germ cells. Hydra is dioecious or monoecious. During summer, interstitial cells in a certain restricted region of the body repeatedly divide and proliferate like reproductive cells, forming gonads, which later differentiate into either testes or ovaries. B. Gastrodermis. The gastrodermis is the inner cell layer of the body wall which lines the hollow and bag-like gastrovascular cavity. It constitutes nearly 2/3 of the entire thickness of the body wall. It is formed chiefly of large, typical columnar epithelial cells with irregular flat bases. The gastrodermis is largely involved in digestion and consists of the following cells. 1. Endothelial muscle or nutritive muscle or digestive cells. These are long, club-shaped, numerous, and conspicuous cells forming the bulk of the gastrodermis. These cells resemble the epithelial muscle cells of the epidermis in all respects except that the basal end is provided with 2 contractile extensions containing a myoneme. This myoneme are oriented at the right angle to the long axis of the body next to the mesoglia, thus forming a circular muscle layer by which animals contract and slowly expand the body. The contractile processes are highly developed around the mouth and bases of tentacles to form sphincters. The free end of a nutritive muscle cell, projecting into the gastrovascular cavity, bears long, whip-like flagella, usually 2 in number, by means of which the liquid food inside body cavity is kept in motion. The gastrodermal cells in the green hydra, chlorohydra, bear green algae, zoochlorella, which give the hydra its color. Nutritive muscular cells may also secrete digestive enzymes into the coelenterin for the digestion of foods. Besides flagella, blunt pseudopodia may also be put out from the free end to engulf the particles of food. The protoplasm of these cells remains much vacuolated in starving hydra. After a meal, however, the cells become gorged with nutritive particles. Electron microscopic studies have shown that the free end of nutritive cells produces microvilli and 2 or more flagella that contain the usual 9+2 pattern of fibers. The apical cytoplasm contains a large number of mitochondria, glycogen granules, secretory granules, and food vacuoles. Both smooth and rough endoplasmic reticulum, free ribosome, and lipid droplets are in abundance in the cytoplasm. Centrally or basally lie nucleus, which includes a single nucleolus. At the base of the cell occur circularly oriented muscle processes containing myofilaments. Pinacetotic invaginations are common at the base of microvilli and membranous vesicles, and channels are present immediately below the plasma membrane. 2. Endotheliogland cells. These cells are smaller than nutritive cells and occur interspersed singly among them. These cells are club-shaped, with the larger end facing the coelenterin. They lack muscle tails at their tapering basal ends, but bear 1 or 2 flagella at their free ends. Endotheliogland cells are of 2 types, mucus gland cells and enzymatic gland cells. The enzymatic gland cells are found in the stomach. They secrete digestive enzymes into the gastrovascular cavity for extracellular digestion. Mucus gland cells are found in the region of the hypostome and mouth. They secrete a slimy fluid serving as a lubricant and also for entangling and paralyzing the prey. The gastrodermis of stalk and tentacles lack gland cells. Gland cells are not under the control of the nervous system, they are independent effectors. Electron microscopy has revealed the presence of many Golgi bodies and a large number of secretory granules, which are functionally divisible into the mucus and enzyme-secreting types. The nucleus is basal, with or without nucleolus and rough endoplasmic reticulum. 3. Interstitial cells. A few interstitial cells occur between the bases of endothelionutritive muscle cells. They may transform into other types of cells when the need arises, i.e., totipotent in nature. 4. Sensory cells. Large sensory cells are also found in the gastrodermis. These cells are supposed to be stimulated by the entry of prey into the gastrovascular cavity. 5. Nerve cells. These are similar to that of the epidermis, but occurs in far fewer numbers. They form a separate, gastrodermal, nerve net on the mesoglia. Nematocysts are absent in the gastrodermis. See mesoglia. Mesoglia, gr, mesos, middle plus glee, glue, of Hydra is a non-cellular, thin layer, sandwiched between epidermis and gastrodermis. It is a continuous layer that extends over both body and tentacles, thickest in the stalk portion and thinnest on the tentacles. This arrangement allows the pedal region to withstand the great mechanical strain and gives the tentacles more flexibility. It consists of a proteinaceous matrix devoid of cellular elements. It serves for the attachment of cellular layers, thus serving as the supporting lamella or elastic framework of the body.